All right, chapter 2 looks at evaluating nutritional information. And this chapter really, more importantly than evaluating nutritional information, really just looks at the research end of it. In chapter 3, we get into how we evaluate nutritional information by grabbing the product there in the store, reading the food label. So in chapter 3, we look at the food label a little bit more. Again, that's evaluating nutritional information. But chapter 2, I don't like necessarily the name of the chapter because it just basically looks at scientific studies. What are those scientific studies? And how do we assess what the heck that nutritional study is trying to convey? All right. There may be some potential essay questions here, maybe explaining some of the differences between some of these scientific studies. That may be somewhat of a good essay question on an exam. Sometimes I ask it, sometimes I don't. All right, so source of dietary recommendations. Our dietary recommendations come from scientific research now for the most part, and that wasn't always the case. A lot of what we understood of what was good or what we should consume came from anecdotal information. And anecdotes is just personal reports. And a lot of what you see written out there is going to be anecdotal in nature. Someone pimping a product, talking about how effective it was for them, how it made them, gain weight, lose fat. You've seen the commercials out there. But what does the science say? And when it comes to a lot of these supplements out there, there's not a lot of great science at all supporting what in the heck they're trying to pimp. So a lot of it is based on anecdotal evidence. Again, just my personal report of what it did for me. And I talk about a lot of my experiences with certain different things, with certain foods, nutrients, supplements, and we'll look at supplements later in the semester. Okay, and again, it's all just anecdotal. It's what happened to me. All right, and when you look at some of the science behind this stuff, it doesn't necessarily support some of these anecdotal things. There's a number of them that look at, this was always given to an individual who always had anxiety, or this was always given to somebody who had an upset stomach. Okay, again, those is anecdotal evidence. All right, when you do the research as to why that works, we can't really find out why. So we use a lot of scientific research now. Make an observation. It's the scientific method. Have you taken Gen Bio? I hope so. If you have, then you could regurgitate this in your sleep. Make an observation. Have a question about something. Form a hypothesis to explain what the heck you think is going on. Review current information out there. I mean, that's how all scientific studies are done now. They take a study and they go, you know what? I like this topic, but I don't like what they did because they forgot to do this. There's some kind of flaw in their design, at least to that person. So they come up with their own study and then just tweak it a little bit. Collect the information, analyze it. What did it show? Share the results. Allow everybody else to try to poke holes in it, yell at you, blame you, all that fun stuff. And then you know what happens? You get to do it all again. And that's what experiments are. Okay? That's what experiments are. Systematic way of just testing your educated guess, your hypothesis. We conduct studies on animal models. We conduct studies on humans. Both have their place. Both have their pluses. Both have their minuses. Okay. When it comes to the animal model, what's predominantly used? Mice and rats are predominantly used. Okay. Why? They're convenient. They reproduce quickly, cheap, gentle, and they're pretty damn similar to humans. Not exactly, but pretty dang close. Okay. This is a great jumping off point to see what we can potentially do, the research that we could potentially do in humans. Okay. There is a lot of studies that get released, done on mice, done on rats, and they are fantastically spectacular. The results are awesome. And because they are very genetically close to us, we assume that that's going to have the same kind of effect here in humans. And so you'll have a lot of fantastical headlines with some of the research that comes out when it is conducted on animals. The bad thing is, closely resembling is not exactly resembling. And there are a lot of potential pitfalls when it comes to applying this find in a human, and it doesn't work at all. We're a walking Molotov cocktail, for gosh sakes. Why mice and rats are used, again, I don't know if I would ask any of these on the exam per se, but if I was to ask, why would we want to use animals? Well, very convenient. They're small. You can house a buttload of them in a small little office. Okay, so and they can be maintained very easily, and they're very calm. They adapt well to their surroundings, so it's easy to use them. They reproduce very quickly. They have a very short lifespan. Okay? Understanding what this does for one individual is fantastic, but being able to observe what that happens over the course of an individual's life, for us, 70 years, 80 years, that's even more fantastic. That lets us really know what the heck this truly does, something that we consume a lot of in our 20s and our 30s, what that means down the road. Excuse me, when we're in our 60s. For mice, they have a really short lifespan of only just a few years, 2 to 3 years. So you can really get an understanding of what this does to their health early in life, in the middle stages, and at the end stages of life. And you can reproduce these individuals very easily. What does that allow us to have? Data. More individuals that we can test, the more data points that we have, and we can truly see if this specific thing that we're testing for really does anything, or if it's, it doesn't have a big, huge effect. And so it's something that we don't really need to worry about. Again, relatively inexpensive, right? Rats and mice are very docile, very gentle to handle. It's creepy that they crawl on you sometimes, but they're very docile in that regard. And again, closely resemble humans, okay? Closely resemble is not exactly resemble, like I mentioned earlier. And so stuff that works fantastically within mice doesn't always necessarily work fantastically within us. Excuse me. We have several different overarching experimental studies. We can have experimental studies where we look at the entire individual, or there's experimental studies that we have where we just look at basically a petri dish. In vivo is using the whole organism, okay? So this is us, working with the rat, okay, working with the mouse. In vitro is experiments on cells, okay? So we want to see how kidneys respond to, what a kidney cell responds to high consumption or high exposure to a certain nutrient. How does a liver cell respond to a exposure to exceedingly high levels of a specific nutrient or whatnot? That's what we're looking at here. So we're taking a look at what the individual cells. So this is more petri dish type of studies. Okay. Again, in vitro studies, those are very controlled. Okay. Very controlled. Very controlled. cells interact with other cells. Systems within the body interact with other systems of the body. You consume food, you consume various different beverages. You're active, you sleep, you go outside and you're exposed to the elements. You're interacting with a lot of different things. So when it comes to these in vitro studies, again, it gives a great jumping off point to do research on the individual. But because they're so tightly controlled and we can control all the variables there, it's a little bit different, because in a person or a mouse even, but definitely in a person, we can't control all those things. And so we're very dynamic creatures. We don't just sit around all the time, very isolated. So in vitro studies, they can be good. They can give us an understanding of what happens. But again, we have to take those with a grain of salt. There was the example of the study done several years ago looking at consumption of grilled meat, charred grilled meat, being linked to cancer. Yeah, okay, they did a lot of those studies, looking at components of the living organism, and then they equated that to the individual. And then they started looking at individuals who identified as consuming grilled food and extrapolated out the connection between higher consumption of charred grilled meat with the development of some cancer-causing agents. What they did not say and did not explain in the headline for that study was, in order to get to this cancer-causing level, you had to consume an exorbitant amount of charred protein, charred meat, to the point where you're not going to be exposed to that amount. You're not going to consume that amount of meat in your lifetime to get to that cancer-causing level. And that's what you're going to hopefully discover when you compare what's portrayed in the general media and what you find in scientific research. To test your hypothesis using one of these two models, an in vivo study or an in vitro study, you have a treatment group. This is the experimental group. This is the group that receives the treatment. You also have a control group that does not receive the treatment. This allows you to see if there was a statistical significance to get that experimental treatment, and if it had a benefit or no benefit at all. If it's statistically meh, then that treatment is not a worthy treatment, and we got to find something else to do to help lower your incidence of certain diseases. Okay, so these are always the two parts of a good scientific study right here: a treatment group, and most importantly, the control group. When we have a study, I made reference to it, there are variables. Variables are factors that can change or that can influence a study's outcome. Okay? We need to control as many variables as possible to assume that the study is accurate. Okay? If variables are not controlled, and this is what makes research in humans very difficult, if you can't control all the variables, that leads some inaccuracies to potentially come out of the experiment, or your conclusions to the experiment can be poked in. Okay? And it can have a number of holes, can have a number of flaws because of an inability to control these variables, or hell, even an inability to even think of that potential variable being an issue. There are a number of studies, and the great thing now about a lot of scientific studies, when you read the research, usually there's a part within the write-up where they even discuss, these are some of the things that I did not think of. These are some of the variables that should be thought of to ways to control for further research down the road. So in a lot of scientific, articles now, they come up with these themselves. But for some of them, they don't necessarily list those, but for a lot of those now they do. So again, being able to control those variables, that's a really important aspect to ensure that the experiment is accurate in what you are assuming is happening. And again, that's what makes it so difficult to do work on us. Okay. Why is it difficult? Again, we're free living things. What I eat is different than what all you eat. What I drink is different what you guys drink. Hell, you guys even eating stuff and drink stuff differently from one another in the room. So it's very different how things interact within the body. Can anybody think of another reason why it would be really difficult to tightly control all the variables in a human besides being free living subjects?
Variables are constantly changing, as are our diets, moods, behaviors. Stress levels can literally affect your body. Age, like Brittany said, stuff like that.
Yep. Those are two. Genetic variety as well. I see is written in here. Yes, genetics play a part into it as well, you bet. Even more simple than that, money. These type of studies done in humans that require tight control of variables. And, when we talk about some of these studies in humans, we're talking about, usually it's talking about, the consumption of a certain amount of carbs or no carbs, or the certain consumption amount of consumption of fats or certain types of fats. And so you really have to regiment meals for all these individuals. Then what do they do during the day? Do they exercise? Okay, now you have to tightly regulate and control that. Are you going to go on the concept of the honor system and have them do it all and hopefully, be able to truthfully write down and do, what they say they did? Or are you going to have it under observation the whole time? So yeah, access to foods. I mean, there's a lot of hurdles, if you will, when it comes to doing research in humans. That's why, when you do read some of these research articles, you're going to think, wow, that's fantastic. And then there's some of you are going to be, holy smokes, what the hell were they even doing? There is so much stuff going on, and they didn't equate for this. They didn't control this. So you'll start to see some of those things. And that's why you can poke holes. And that's why we have all the bickering we do when it comes to nutritional research. Animal studies have some limitations. Again, like I kind of mentioned earlier, there are some slight dietary differences, some slight metabolic differences, physiologies. There are some slight physiology differences when it comes to animals and humans. So that's why it's a great jumping off point, animal research. But when it comes down to it, it may not be exactly what's going to happen in us. So it can help guide human research. And that's the great thing about doing research on animals.